Okay. Now we had talked that centrioles get duplicated during the S phase, they move towards the poles during the prophase. What is the function of these centrioles? Centrioles are microtubular structures. What is their function is that they are going to form microtubules only, but these microtubules are going to solve the purpose of separation of the chromatin material. How? They are going to form, important term, please remember it now itself, spindle fibers are to be formed. Spindle fibers are to be formed so that the chromatin material gets separated. Now, over here the prophase has completed, centrioles have moved to the poles, the chromatin material is condensing to a level at which we actually want it to condense, and at the same time spindles have been started to form. Somewhat primitive spindles are being formed. Now what happens in the next stage is that these are completely condensed. The stage where I and you, when we are looking under a microscope, can see clearly the chromosomes is known as metaphase. What happens in metaphase is that all the chromosomes, whichever are present inside the cell, arrange themselves in the center with the help of spindles. This is just for understanding purpose I am making it. This is the centromere in each case where the chromosome is present. These are the sister chromatids. One being supposedly this is having this coding, so it would have similar coding for DNA, similar DNA content, because this the other sister chromatid has nothing else but the duplicated DNA content. Centrioles have their spindle fibers coming like this. It's like this. Alright. Now there is a pad-like structure which is present at each centromere known as kinetochore, over which the spindle comes and attaches itself. Kinetochore is present on each centromere, both the sides. The spindles come and attach themselves at this position known as kinetochore. Now it is something like Spider-Man picks up his stuff. You must have seen, or if you haven't seen, what he does is he throws his fiber like this and then he pushes back the stuff wherever it is kept. Same way this spindle would come over here, it would attach itself and then it would retract itself. Alright, now this retraction would happen in such a way that the centromere would split. That is part of the next stage, but for once what you need to remember in metaphase is that metaphase have metaphasic plate being formed where all the chromosomes are arranged in the center of the cell, exact center. Now there is no nuclear membrane, so cell is a uniform thing that we are considering over here. Chromatin material is uniformly divided or scattered inside the cell in the prophase. Then that scattered material gets more condensed, it comes in the center and metaphasic plate is being formed. So this stage is characterized by the metaphasic plate. Now what happens is, I give you example of that Spider-Man thing. This spindle is to be retracted and as a result the centromere breaks. Now what happens, this one spindle takes along one of the sister chromatid and the other takes along other sister chromatid. This is going to happen in the next stage. Alright, so you see the sister chromatids got separated from each other, each is going towards the different pole. Now over here, those who have studied cell cycle or cell division in their previous classes with in-depth knowledge, they can make it out that this is the stage where the chromatin material is on the verge of separation basically, because one has gone to one end. Now the spindles would retract further. It's like you throw some thread over on something, then you push it back. So they are moving towards the pole. When they have moved towards the pole to such an extent that it is clearly visible that the chromatin material has gone to opposite poles with each sister chromatid, it does look like fly, but please remember these are the sister chromatids only that we are drawing over here. When they have gone to the poles, opposite poles, that stage is known as anaphase. So the points that are to be remembered during anaphase is spindle retraction, which takes place towards the centriole. Alright. Now, how do we identify that telophase has started? These two stages are very clear in to observe basically in a microscope. Under a microscope, it is very clear to see the metaphase because metaphasic plate is being formed. It is very clear to see the anaphase because they have started moving towards the poles. This broken sister chromatids which are separated from the centromere because the centromere splitting has taken place. So this stage would be characterized by centromere splitting, and it would be characterized by the movement of sister chromatids towards the poles. What happens in telophase is that these sister chromatids would come together and they would again somehow decondense themselves. They were condensed in chromosomal form. They would decondense themselves, and all those things which were degenerated or which disappeared during the prophase, they would start coming back. So what disappeared in prophase was the nuclear membrane, and when the nuclear membrane would come, other endomembrane structures like endoplasmic reticulum and Golgi bodies would reappear, and chromosome material would get back into its chromatin form. So you get two nuclei by the end of telophase, and of course other organelles which are required, they have also duplicated themselves during the G1 phase. So you have a sort of separation. The organelles for this cell, suppose this is organelle 1, organelle 2, organelle 3, they also arrange themselves over here, and this is organelle 1 dash, organelle 2 dash, organelle 3 dash. So there is a clear cut separation. As you can see, you are having two nuclei, daughter nuclei, which are being formed, but they are present inside a single cell for now. So now what has to take place? The cytokinesis has to take place because our karyokinesis is complete. So you see there is so much of activity which is taking place in the karyokinesis. Now what happens in cytokinesis is that, considering this as one daughter nuclei and this one as other daughter nuclei which are being formed, now there has to be a separation. If the separation does not take place, then it is multinucleate condition, and remember the term, we use syncytium for this. Multinucleate condition occurs when this activity is not followed by the division of cytoplasm. Now we have two types of cells that we consider for cytokinesis. We don't get into fungal cells and the rest ones, five typical types of cells. All we talk about is a plant cell and an animal cell. So in case of animal cell, what happens is that a furrow starts developing like this. Alright, this furrow deepens further. You might have somewhere or else in your life drawn a finger through a droplet of water and seen that that drop divides into two. Same thing happens over here. This furrow is going to get further deeper, and at one stage this would come and join this, and this would come and join this. So the two daughter cells would be formed. So this is cell furrowing, which is seen exclusively in animal cells as a method of cytokinesis, that the furrow starts from the periphery and gets deepened and deepened, and in the end it meets, and we have two cells which are being formed. But when I talk about plant cell, it won't be that easy. Why? Because plant cell does not have a simple cell membrane as in the case of animals. It is surrounded by a cell wall as well, cellulosic cell wall. I hope you know that. If you don't know that, just remember that cellulose is a complex carbohydrate which surrounds the cell membrane of a plant cell, and that is why plants are not that much able to move themselves. We are blessed with the ability of movement and locomotion because we don't have cell walls. It is a rigid carbohydrate. So what happens in this case, that in any case the furrow will not result in division of cell wall as well. So instead of developing a furrow, few phragmoplasts come and arrange themselves. They form the vesicles, and a cell plate is being formed in the middle. Alright, this cell plate is formed when calcium pectate comes over here. And if you remember that what is the purpose of calcium pectate in a plant cell, it is the formation of middle lamella. Now what is middle lamella? It is like, you might have seen that in a construction or somewhere, these are the two bricks and cement in between, and then we have other brick, cement is present over here. Same is present in animal cells as well. So this middle lamella is the cementing of calcium pectate, which has cellulose over here and cellulose over here. This further broadens and reaches the ends, and we have two different cell walls which are present, and as a result two different cells would also be there. It would further lead to generation of plasma membrane inside both, and we have two different cells which are formed by cell plate method. What you have to remember is the phragmoplasts would come and assemble themselves in the center of the cell for the cytokinesis in plant cell, because these phragmoplasts would further form the vesicles. So the term that is to be remembered for cytokinesis of plant cell is the phragmoplast. Its assembly, its formation of middle lamella, and the generation of cell wall which needs to be present in each cell. So we have two daughter cells which are being formed, which have their own cell wall, and it is the result of cytokinesis. Now you get to know what is M phase and which division is responsible for growth in all the organisms. It is the mitosis, which is manifested in two stages, first being the karyokinesis, second being the cytokinesis. Major activity of cell division is done during karyokinesis. It has four stages: prophase, metaphase, anaphase and telophase, which is followed by cytokinesis, and in the end we have two similar, identical I must say, to the parent cell